Hi, my name is Emma Cartney and I'm here to introduce you to the ERGA Pilot Project and the Pilot Committee's activities to date. This committee is coordinated by myself, Alice Mouton, Giulio Fermenti, and our contact details here are at the bottom of the screen. So the goals of the pilot project were to sequence and assemble and annotate at least one high-quality reference genome for a species suggested by each ERGA-associated country to test our distributed and trans-border model of reference genome establishment. This project was initiated last year by a survey distributed by the ERGA Samples Permits and Processing Committee, where each country could suggest at least 3 feasible species to be sequenced and assembled throughout the pilot. So at the end of this process, we had 95 feasible species represented and suggested and moved forward for the pilot project. And these were supported and championed by 95 genome teams. So each one of these species has an associated genome team. And this is made up of several roles. And we really promote that each of the roles within a genome team are trans-border in nature, so that we are promoting collaborative trans-border partnerships within ERGA. Each one of these reference quality genomes will meet the Earth Biogenome standards. What this means, to do this, we have ensured that each species has the necessary data types to do this, and that being Pacific Biosciences or Oxford Nanopore reads, and supplemented with Hi-C data, and our annotations then carried out using Illumina RNA-seq data and or Pacific Biosciences ISO-seq data. We wanted to again ensure that each one of these genomes and annotations that we were generating had the associated metadata collected. To do this, we partnered with the Samples and Permits Committee again, and we produced our metadata manifest, which each genome team had to complete and upload to our metadata brokering service, who we partnered with, known as COPO. COPO would then broker this information into the ENA, our public data repository partners, and that metadata would then sit alongside each of the pilot project genome assemblies. So our milestones that we have reached today, of the 95 species, 20 species are ready to sequence and are in sequencing centers. 18 more species are actively being sequenced, and 28 additional species have now data associated with them and are being assembled. Again, this is a grassroots effort, and much of the funding required for the pilot project has come from the sample ambassadors and the genome teams themselves. If you would like to hear more from them, I would really encourage you to log on to this YouTube clip where you can hear them speak about the ERGA Pilot Project in their native tongue. I would like to also thank our committees. Each one of these committees have really strengthened and bolstered the success of this pilot project so far. I would like to thank our sequencing centers who span across Europe and who have been wonderful participants in the pilot project to date and will into next year too. And our sequencing providers who have sponsored data or guidance and consumables for the pilot project. Finally, I would like to thank our council and our chairs who've really guided and directed us into making the pilot the success it is today. And if you would like to join us, please, I encourage you to email us.